Yeah, so thank you very much for inviting me. And I really love the place and the vibe of the place. And my room actually is amazing. So thank you so much. Yeah, really, I feel as if I am an Oxford Fellow or something. So yeah, it's amazing. Thank you very, very much for inviting me. So today I'm going to discuss about some new results we have on some novel phages that we isolated based on clinical isolates. And using the knowledge that we have from previous publications in the lab and previous work in the lab, we now want to see whether we can genetically modify these novel phages to make them target intracellular bacteria, intracellular E. coli. So why are we bothered to do that? For me, when I first started my interest on phages, that was via a BPSRC fellowship in 2016. I was not really sure of what I'm doing. And then I thought at the time, can we use phages to tackle the problem of AMR? And you can see from this report of 2019 that the problem of antimicrobial resistance is really, really serious. And if you notice the numbers here, very many people are dying still because of the problem of antimicrobial resistance. So could phage therapy be the solution? And this is a really, really lovely paper published very recently from Jean-Paul Prinot and colleagues. And what they did, they took the personalized cases, that, like the clinical trials, and they analyzed them retrospectively. And they found that indeed, phage therapy can be the solution and is successful. I know it's tricky for us to gather everything, and I had the chance to chat to him very briefly in the Malta conference, and he said it was really huge work. But I think it's really, really promising for all of us who are in this room, at least. Yes, so one good thing with phages currently is that in the meantime, synthetic biology has developed, and what we can do now, we can safely and easily genetically modify bacteriophages. This is mainly due to their small genome, and we can give them the preferred characteristics to then use them for downstream applications. And of course, some applications relate to biocontrol, biodetection, but also therapeutic applications. And this is a review that we wrote for my lab. My postdoc led it, Jessica. And here we summarize different ways via which we can genetically modify bacteriophages. And of course, you might have heard about changing the tail fibers. Why? Because the tail fibers give this specificity between the phage and the host. So by changing the tail fibers, we can broaden the host range of the phage. And of course, other ways is to change the DNA with addition of genes and enhance the payload of the phage so that we make a phage that can target other elements and not only the ones that is designated to target. And you might have seen loads of paper recently on making phages from lysogenic to lytic, mainly because when we do phage therapy, we are only allowed to use lytic phages and not lysogenic. And then, of course, there are other very nice papers. I know there are some from Rob Laville's lab where they did directed evolution. They evolved phages, they engineered phages to make them safe and very efficient to be used for patients. And for me, seeing on the literature the use of engineered phages to save lives, it's amazing. I'm very, very happy about it. And these are just, there are many, many papers around, but these are some very recent examples. So this is an example from Hatful Lab in collaboration with others, where they did this evolution of phages. They evolved phages, and then they picked the best ones, and they made the cocktail, and they cleared the infection of a cystic fibrosis patient who had the Mycobacterius abscessus, and that enabled them then to do transplantation successfully. And this is another very interesting paper in which we also wrote the news and views from my lab. This is led by a very big group in Copenhagen. And what they did, they selected phages from cancer patients suffering from hematologic cancers infected with E. coli. and then they genetically modify their tail fibers to carry CRISPR-Cas, and then they picked the best ones. They made a cocktail, which they named SNIPR001, and this is now on clinical trials. It's the first case of genetically modified phages being on clinical trials. And the good thing with that is that you can give a chance to immunocompromised patients, for example, patients with hematological cancers, that they cannot stand antibiotics. This phage cocktail is efficient and it's shown to be safe for such patients to clear their infection. So now I have to very briefly talk to you about previous work in my lab, because what I'm going to present is a follow-up of what we have found so far. So when I first started my lab, we modified phages to make them fluorescent. And I don't know if that is working. So we could see that, we could see that upon tagging phages with fluorescence, they would gather into clusters and they will invade human cells. This is the human cell, this is the nucleus, this is the periphery of the cell, this is the phage cluster. They would go inside vacuoles at all times. We tested that in 2 different cell lines and we found that to be the case. And we further checked that and we found that phages are invade human cells via phagocytosis. They are degraded via L-cysteine phagocytosis. They do not activate autophagy, so they are not regarded as an enemy. And when we tested inflammatory markers by qPCR, we found that in the presence of the phage, there is no increase. So phages, for some reason, they invade human cells randomly and they are degraded. And this was published in 2018 and 2020. Because of this observation, we thought, what if we become a bit more clever and we now genetically modify a phage to express something that is essential for human cells? So we modified further our phages to express EGF. Epidermal growth factor is an important nutrient for human cells. And the idea was to increase the affinity between phages and the human cells so that phages enter even more human cells and hopefully clear even more intracellular infection. I have to mention here that we noticed before, even in the non-modified phage, that they could clear intracellular infection to an extent, not 100%. But the idea was, what if we do that and we make that even better? So indeed, this is our GFP phage and this is the GFP EGF. I have to say here that for all these studies, we focused on K1F. K1F is a T7-like phage that expresses endo-alpha cellulase on the tail fibers and can target E. coli K1. Up to now, we have been using EV36. EV36 is a hybrid between E. coli K1 and E. coli K12. So we were mimicking the pathogen, but in reality, when we were in the lab, we were using something safe, mainly for us to do microscopy and for the students. So here you can see that the phage that expresses EGF is accumulating much more compared to the GFP. And also, when tested in 3 different cell lines, this is urinary bladder, this is brain human cell, and this is skin fibroblasts, you can see a significant increase in the ability of internalize human cells compared to the only GFP phage, indicating that this phage is working. Importantly, this phage now is able to clear more efficiently intracellular bacteria compared to the only GFP. And especially to epithelial cells where it invades even more. So you see that so far, the idea of creating a phage that can target intracellular bacteria seems to work well. I think I mentioned before very briefly that we noticed that phages enter randomly via phagocytosis and is degraded via L-cysteine phagocytosis. But now, with our EGF phage, we noticed this dotted pattern. And this dotted pattern looks like a lot, of course, with EGF receptor, but also with endosomes. So we now wanted to check why is this phage more efficient on entering human cells? Could it be because now it can also enter via endocytosis? And when we co-stained the phage with early endosomal markers, we could see that this phage now is also able to enter via endocytosis, and this is why it makes it more efficient on entering human cells. So this was published in 2023, and this is what we see. You genetically modify phage, and you see increased cellular uptake. So now I have to go to my new results. Yeah, yes. So now, what we want to do, we want to combine all our knowledge together for clinical purposes. This time, instead of targeting EV36, we are targeting UTI-89, which is a clinical pathogen. Our aim is to find novel phages towards this pathogen and also test if these phages are good for pathogen control and suitable in terms of safety. So what we did, we isolated, we collaborate a lot with the hospital, so we isolated phages around the area of the hospital. My students gave all these funny names, and I'll just let them play. I don't mind. Initially, bean and avocado, yeah, I mean, my students, bean and avocado, they were always together, and we couldn't separate them. This is because if you notice, all of the other phages are K1. So you see that the tail here has this endo-alpha sialidase. It targets K1 pathogens, whereas the avocado is a T4, T4-like. So for some reason, these two together worked amazingly, but we couldn't really separate them. After a lot of effort, we managed to separate them, and this is how they look like. And then we sequenced them. I'm very bad on bioinformatics, so I had to ask for help. So Luis Daniel Rodrigo Melo helped us with analysis. And you can see that K1F has been used in all cases as a control kind of to compare, because all our previous work was on K1F. And you can see there's great similarity with our novel phages, of course, except from avocado, which is the T4-like phage. For example, lemon and lime, 99.4%, but lime does not have the GB2. And then jalapeno has another tail fiber more than shorthead, which you will see later, it helps. But they still have a very good similarity among them. So we have done different assays now to study these phages. We have done adsorption and one-step growth curves. And you can see that the short prit and the jalapeno are the most efficient at binding, and the bean was the least efficient unabsorbed compared to the rest. And also, you can see here that bean has the largest eclipse period and took the longest to initiate the infection. But all of these phages were able to target UTI-89, which was not the case with K1F, which is a good thing. And they all seem to be K1 specific. Now, what we did later, because we collaborated with the hospital, they gave us from the hospital E. coli coming from infected blood from patients. And we thought, let's try all these isolates to our new phages. And of course, we knew that it was not going to work for everything, because some of the E. coli isolates are not K1. They have other antigens. But still, if you can see here, bean was the most effective in clearing even the random clinical isolates from our patients. And then when we sequenced the clinical isolates, we could see that even though almost all of them, they had K1 antigen, some of them had also O antigen and H antigen and other antigens, and that could probably explain why our phages, who are isolated based on a K1 strain, the UTI-89, cannot be fully effective. Of course, they all have AMR genes, and we couldn't identify all the sequences so far. Yes. So we are now in the process of this, for some reason, didn't, it should show here, but we're now in the process of analyzing with Padlock the different defense systems that we find on our isolates. We have found so far Lamasu, Sophic, RM, different defense systems. Importantly, the strain that we had, the UT89, was a lab strain and didn't have any defense system. So we are very pleased to see that the phages that were isolated are able to clear infection even on isolates that have some defense systems. Okay, because I'm running out of time. So now we are in the stage where we want to analyze LPS. We want to also understand how the receptors are working, what is going on, why our phages are effective to some of the clinical isolates and not to others. We want to link robustly the genotype with the phenotype, and ideally, we want to select over the best phages and then modify them with GFP EGF to make them able to clear intracellular infections now on a clinical isolate and not only in the lab. Okay, so with that, I want to thank my lab, especially Josh and Jess, and thank you very much for your attention. Thank you.